Hello, general botany students. It is me, Stephanie, and welcome to this week's virtual lab that's going to be all about cladograms. Remember from this week's lecture video that cladograms are hypotheses. We use them to infer evolutionary relationships, and since it's pretty difficult to prove with hard evidence how things evolved, since they didn't fossilize very well and no one was there to witness this evolutionary history, we must consider evolutionary relationships as hypotheses. Hypotheses are predictable, falsifiable statements. These are the primary features of the scientific method, regardless of the scientific field, right? Physics, chemistry, biology, we all use hypotheses and want to test them. Phylogenetic analysis, also known as cladistics, is a discipline that infers evolutionary relationships between living and extinct taxa in order to learn about patterns and processes of evolution. This branch of biology also tests hypotheses. For example, species 1 and species 2 are more closely related to each other than either are to species 3, or species 3 evolved before species 1 and species 2. Both of these statements are falsifiable. So in order to reject or retain a null hypothesis about evolutionary relationships, we must construct a cladogram. This will be the main focus of today's lab. People make cladograms based on morphological and macromolecular characters for the species that you're interested in. And they view these characters in the form of usually a large matrix. This data matrix will allow you to find your tree type topology. Tree topology is another way to say the branching pattern of your cladogram. So this data matrix will allow you to find the tree topology that represents a model of how the taxa in your group of interest have evolved over time. So consider this simple example here. Here are two competing hypotheses showing two different patterns of relationships between the same set of three taxa. Notice that in one, there are many, many more changes overall, and in the other, far fewer changes. Well, let's see this classic example of parsimony analysis using a well-known set of organisms. We've got bats and bunnies and sparrows. Actually, now the truth is that you could replace bunnies with really any mammal here. So like bats and humans and sparrows would also totally work here, because there is a well-known, actually, evolutionary relationship between bats and other mammals, namely that bats are mammals. They are warm-blooded things, which sort of comes as a shock to people since they have so many similarities with birds, right? Birds and bats can both fly. They've got these wings. Very cool. Okay. And the truth is that bats have a ton in common with bunnies, even though bunnies don't fly. So bats and bunnies, they both have, for example, mammary glands. They both have fur. They both are warm-blooded. They've got these similar, very fancy inner ear bones, very similar skull forms, and many other traits, actually. And so that's what's represented by the red dashes here. And these are all traits that are shared between bats and bunnies. Notice that none of those red dashes are on the way to the bird lineage, the sparrows here. Well, it's because the sparrow doesn't have those fancy ear bones and a particular skull and warm-bloodedness and mammary glands and fur. No, birds don't have any of those. In fact, the only thing that a bird has in common with a bat is the wings. And so with these two different cladograms that are actually showing the exact same information in terms of what morphological characteristics our three taxa have, they're showing completely different information based on what the relationship pattern is between these three organisms. And the way that a parsimony analysis works is we want to pick the simplest option. We want to be as stingy as possible. We want to pick the cladogram that is the most believable. And it's much harder to believe a scenario where warm-bloodedness and fur and mammary glands, etc., evolved independently twice. All of those things would have had to evolve independently twice on the way to bunnies and on the way to bats if bats and sparrows are more closely related to each other than either one is to a bunny. So clearly here, the cladogram that has bats and bunnies more closely related to each other, this is the simpler of the two cladograms because it has fewer total changes, and most importantly, it has fewer homoplasies, fewer instances of convergent evolution. And so it is more believable, and so it sort of wins the parsimony analysis, and we believe the scenario where bats and bunnies are more closely related to each other than bats and birds are. Okay. But this is, of course, botany. And so let's talk about a botanical example here. So here we have 3 conifers. All 3 of them produce cones, although you can't see them in all of these pictures. Just know they all produce cones, and they all have a lot in common. Notice they're all photosynthetic. They produce leaves. I'll tell you some other stuff. They're woody. They're trees. They have a lot in common. And so when we see something like this, a situation where we've got these features that are held in common between multiple plants, multiple organisms, we might want to ask a question about their relationships with one another. And so here I have kind of asked a couple of questions, and actually I have some theories about them. So my hypothesis is that conifer 1 and conifer 2 are more closely related to each other than they are to conifer 3. So in other words, 1 and 2 are more closely related to each other than 1 is to 3, or than 2 is to 3. We always have to produce a null hypothesis for any hypothesis that we produce, and so the null hypothesis is all other scenarios. And in this case, the other scenarios would be, well, 1 is more closely related to 3, or 2 is more closely related to 3. In fact, there are really only those 3 possible cladograms in terms of showing the relationship between 3 organisms. Well, actually, I have another hypothesis about these 3 conifers. My other one is that conifers 2 and 3 are from an older lineage than conifer 1. In other words, they sort of evolved before the lineage that led to conifer 1. And the null hypothesis for this would be, again, all other options. So maybe the other option would be that conifer 1 is from an older lineage than 2 or 3, than conifer 2 or 3. Okay, well, in order to answer either of these questions that we have, we need to build a cladogram, because that's what tells us, that's what shows us the relationships. And when we know the relationships, we'll know the ages of things, maybe not the exact age that they evolved, but at least the relative times that these 3 organisms evolved relative to one another, when their common ancestors existed relative to one another. Because, right, they're all technically the same age. They are all existing right now in 2020, but they have different ages in terms of the length of the lineage that they came from. Okay, so if we want to build a cladogram, because we have hypotheses about organisms, this is how we do it. We have 6 steps. Anyone constructing a cladogram must go through the following steps. Number 1, select all of the taxa in your group of interest. Okay, the more taxa, the more complex your cladogram will be, by the way. Number 2, step 2, choose an outgroup for your group of interest, if possible. This is ideal because it will allow us to polarize the traits and deem them either ancestral or derived, which will be very useful when it comes to making the call on some of these questions that we have about relationships. Step 3, select and define the characters. Again, you want as many as possible, but the more you get, the more complex. But the more characters you get, the more confident you will be with the hypothesis that you end up retaining or rejecting. All right, step 4, define character states for each taxon, including the outgroups, and the character states for all of the outgroups will be zeros. So remember, we've kind of got these 2 things. Notice I've got characters listed in step 3 and then character states listed in step 4. Well, characters are sort of the broader category, and then character states are the specific. So maybe an example would be a character is petal number, and the character state is 5 petals. Or character is petal color, and the character state is yellow petals. Okay, moving on. So step 5 is to perform a parsimony analysis to determine the simplest possible cladogram out of all possible cladograms. That's going to be the main point of today's lab. And then step 6, interpret your most parsimonious cladogram and assess your hypothesis. Either accept or reject your null hypothesis. Okay. So let's use our example here. Let's go through those 6 steps. Step 1. Here are the 3 conifers. These are the ones that we've been talking about. These are the ones that I'm interested in their relationships. Okay, step 2. We have found an outgroup here. And by the way, outgroups will be provided for you in this class. This is an organism that's not closely related to our group of interest. We know that it is from a more ancestral lineage than our group of interest. And we need to really look at the traits of our taxa because we're going to be using them in a cladogram. And we need to kind of know if these traits are going to be derived or ancestral. And so the outgroup will help us answer that. Step 3, we've looked at them. Let's say we're out in the field, we've looked at these conifers, and we have figured out that there's some variation between these conifers, namely in these characters. The presence of seeds, the presence of a thick cuticle, the presence of linear leaves, and the presence of serotinous cones. Serotinous just means they only open with an environmental cue. They stay closed at maturity, by the way. Doesn't matter what it means right now. That's not the point of this. Moving on, step 4, we need to actually analyze those traits for each taxon. And here we've done that. Notice that all of the outgroup's character states are zeros. These are the ancestral ones, and so the outgroup must have the ancestral condition for everything. So all of the other zeros on this matrix that are not part of the outgroup's row mean that that conifer has the ancestral condition for that character. All right, and now for step number 5, we are going to need to draw multiple possible cladograms and then determine the simplest out of all possible cladograms once we've traced the character states out onto it. So here we go. Here the O stands for outgroup, and I'm drawing all 3 possible trees. If there are only 3 taxa in our group of interest, then either taxon 1 branched off first, in which case 2 and 3 are more closely related to each other, and since they're most closely related, it really doesn't matter which order we put them in here. But the other 2 cladograms here either show 3 branching off first or 2 branching off first. These are the only possible orientations that these 3 taxa can have with one another in a cladogram. Look at this very first character. Notice that there is a derived character state for all 3 taxa. And that means that on all 3 of these cladograms, we're going to have this character represented in the same place, right? At the base, in between the outgroup and the first species that evolved. A trick to this. We don't really need to map every single trait onto every single possible cladogram. We can just compare the conifers to the outgroup, and we need to see which one has the most in common with the outgroup. That's going to be the one that is most likely to have evolved first, and in this case, it's conifer number 3. It has 3 similar features with the outgroup. Only one of those characters is derived, and that's seeds. The thick cuticle, the linear leaves, and the serotinous cones are all absent in both the outgroup and in conifer number 3. And that's why, actually, this is evidence that that top right cladogram is the most parsimonious, which is why it was just circled in yellow. This is the one that shows the conifer that has most in common with the outgroup, kind of closest to the outgroup. And now we're just mapping on the rest of the traits, both Linear leaves and a thick cuticle are present in conifer 1 and 2, but not in conifer 3 nor the outgroup, and so they would be situated right there in green. But serotinous cones is only present in taxon number 1, the conifer number 1. So this is our completed, most parsimonious cladogram. Most parsimonious cladogram has 4 total changes, as represented by these 4 colored hash marks on the cladogram. For consistency's sake, I want to prove to you that these other 2 possible cladograms, these other 2 hypotheses, were indeed less parsimonious, meaning they were less simple. They had more homoplasies. Notice that for this one, if conifer 1 branched off first, then the traits that are present in in conifer number 1 and conifer number 2, they would have had to evolve independently twice, since conifer 3 did not have those states, those character states. So in both of these cladograms, both the linear leaves and the thick cuticle would have ended up being homoplasies. And of course, conifer number 1 still had those serotinous cones. And the seed was still basal to all 3 of them. And so actually these other 2 possible cladograms each had a total of 6 changes. More changes, less simple. Just simply more changes is more complicated. It implies that there was more convergent evolution, which is less likely. And so these other 2 possible cladograms, even though they are relatively similar to the winning cladogram here, they're slightly more complicated and therefore less parsimonious, and they have sort of failed the parsimony analysis, while the cladogram in the upper right has passed the parsimony analysis. It is our winner, if you will. Right now, I would like for you to answer a few questions on your own, please. For the small monophyletic group that is conifer 1 and conifer 2, name a synapomorphy. For the same group, name a simplesiomorphy. Now, I want you to find an autapomorphy on this cladogram. This is a new word. Aut, A-U-T, stands for self, and apo sort of means apart, morphology. And so there's one trait on this cladogram that represents an autapomorphy, and it means it's a trait that is only found in one organism. It sort of sets that one apart from others. And notice that it's the serotinous cones trait for conifer 1. That is the one autapomorphy. The next thing I want you to do on your own, because I want you to sort of have this redrawn in your notes, is I want you to list all monophyletic groups possible from this cladogram of all the 4 in taxa, including the outgroup. What are all of the monophyletic groups? Ask you the sister group of conifer 1. What would you say? Conifer 2, hopefully. That's the closest relative. It's the only other descendant of the common ancestor that's closest to 1 and 2. But what about the sister taxon of conifer number 3? Or actually, in this case, it's a whole group. We need to go down to the most recent common ancestor of 3 and then sort of take the other lineage. And so actually the sister group of conifer number 3 is the group 1 plus 2. Cool. Homoplasies on this cladogram? Remember, homoplasies are instances of convergent evolution. I don't see any. To go back to those original questions, a synapomorphy, we've got leaves and cuticle for 1 and 2. And a simplesiomorphy for them would be the presence of seeds. Are there any non-monophyletic groups that you can imagine on this? Well, there's a monophyletic group, 1 and 2. What about this group right here, 1 plus 3? Yeah, that's non-monophyletic. Here's another non-monophyletic group if we were to put 2 and 3 together. Good monophyletic groups include all of the descendants from a single common ancestor. Remember our hypotheses from earlier. Back to step 6 now. And we can definitely reject the null for hypothesis 1 and get some evidence for our alternative hypothesis number 1. Conifer 1 and 2 do appear to be the most closely related, and there's their most recent common ancestor. There are no other descendants coming from it, and that's why they make a monophyletic group, and they are kind of each other's sister species, at least relative to conifer 3 on this cladogram, which is a more distant relative. Now, hypothesis 2 was a bit more complicated. It said conifers 2 and 3 are from an older lineage than conifer number 1. Time is increasing from the bottom to the top of this cladogram, and so the most distant common ancestor is the one that is shared with the outgroup. And here, the second most distant common ancestor, there's only one descendant from it, and it is conifer 3. And so conifer 2 is from a more recent lineage than conifer 3. It's from the same recent lineage as conifer 1. And so we can accept the null, actually, of hypothesis 2. Here's a different example. This one might seem a little more difficult because we're kind of starting in a different place. Here I have given you a data matrix, and we've got a bunch of information. We don't even know what these plants are. I've never talked about them before. There's some stuff we've never talked about, like stamens, but that's okay. All you need to know is that we've got a matrix here, and we can use this information to build a cladogram. Okay. So we've got this sort of imaginary group of organisms, actually. They're in the genus Botanica, and we've got 5 species. Botanica alba, Botanica lutea, Botanica nami, Botanica purpurea, and Botanica rubens. And each one of these 5 species has a character state for these 1, 2, 3, 4, 5, 6 characters that are on the very top row. Okay. Well, I've got some hypotheses about these. Just these hypotheses, I'm just kind of making them up for our example based on this matrix. Maybe my first hypothesis is, or actually my hypothesis in this example is that species B, purpurea, so Botanica purpurea and Botanica rubens, are more closely related to each other than they are to any of the other species, namely Botanica alba, Botanica lutea, and Botanica nami. Notice I've just reduced Botanica to the letter B on the slide here. That's okay. So what I'm saying is the last 2 species on this, and the last 2 rows on this matrix, Botanica purpurea and rubens, are more closely related to each other than either of those are to any of the other 3 species on this matrix. Okay. So if that's not the case, the null, then the null would be true. And the null here are all of the other cases. So we could state that as this. Species Botanica purpurea or Botanica rubens are more closely related to either Botanica alba, Botanica lutea, or Botanica nami than they are to each other. Okay. What we're going to do here is start at step 5. Because we have been provided a matrix, and so we can kind of skip through steps 1 through 4. We have essentially done that, let's just say, and here is the matrix. Or let's say somebody else did that, and we are the researchers here, and we need to determine if the information that these folks have collected can be supported by our hypotheses. Okay. Or can support our hypothesis. So here are the procedures for testing a phylogenetic hypothesis. We're going to build a tree using parsimony analysis again. Okay. Now, the first step here, since we don't have that same type of data matrix as we had previously from step 5, is we're going to convert all of these wordy words into zeros and ones, into these binomials that are sort of easier to interpret and easier to, I think that they're kind of easier to build a matrix, a cladogram on. Because I think that it's just kind of an easier way for us to compare derived versus ancestral states in these different taxa. And so the upper cladogram here is our original one, and then the lower one, I've just reduced it to a code, but it has the exact same information. So we still have those 6 characters across the top. We have those 5 species of interest on the left. I've reduced them to A, L, N, P, and R for those 5 species names, respectively. And we still have our outgroup on that top. And then the zeros and ones represent ancestral states, while ones represent derived states, and this is all relative to that top row of zeros, the outgroup. So now we need to build a cladogram, and this is a much bigger matrix than the one that we had just done together with the conifers. So first we need to find when we have this matrix, what we need to do is first find the taxon, here, the species, that's got the fewest derived traits. So in other words, this is the one that is most similar to the outgroup. This will, in theory, be the one that most likely branched off first. The one that has the most in common with the outgroup, okay? The next step will be to find the species that is the least derived from the taxon that you just determined from step 1. This will be the taxon that branched off next. And you kind of continue this process of seeing, well, which one is most similar to the one that just branched off? You continue this process, like it says in step 3 here, continue this process until you've mapped them all out onto one cladogram. And if there's ever a tie, like, oh, these both they both have 3 derived character states compared to the previous taxon, well, then we need to assess the rest of the cladogram and the rest of the taxa, seeing if there's any evidence for a smaller group that will kind of help us build this simple cladogram. And so let's see an example of this using our zeros and ones. Know that the outgroup branched off before the other 5 taxa on this cladogram, but we don't know what order these last 5 branched off from. We don't know who's most closely related to whom out of the 5 that are not highlighted, not highlighted right now. And now that I'm erasing the highlighted, we can see the outgroup. Well, remember, our first step is we need to find the taxon that is most similar to the outgroup, that is the least derived from the outgroup. And so notice that species A, it has 5 ancestral character states. The other ones have 4, 2, 1, and 0 ancestral states. And so it looks like species A is sort of our winner here. It's the one that most likely branched off first before these other 4 species, at least. And so let's sort of redraw this cladogram. Next, we need to repeat that process, but for this smaller group of species, that is the other 4, that we need to figure out their relationships with one another. L, N, P, and R, apparently. And so now we need to figure out which one is sort of most similar to A. Which one is sort of the least derived from A. And so we need to sort of restart here when we're keeping track of the similar traits to A. Has 4 of the same ancestral characteristics as A, N has 2, P has 1, and R has 0 of the same ancestral states as A. And so it's likely that L was sort of the next was part of the next lineage that branched off after A. So let's add L. And now we need to look at the other three. We've got N and P and R. And we need to sort of decide, are these going to be, are any of these going to be more closely related to L than they are to each other? If so, they will need to be branched directly off of that L lineage. Well, it looks like N also has two ancestral characteristics that are shared with A and L, and so it's likely that that one branched off before P or R, but it also has features in common with P and R that are not shared between A and L, and so it is probably going to be in a group with P and R as opposed to with L. Now that we have a likely scenario for how all of these taxa might have evolved from one another, let's map out all of the traits onto this cladogram. Remember, in a real parsimony analysis, we would be comparing the total number of trait changes on this cladogram relative to other possible cladograms of these 5 species. So it looks like trait number 1 is the only trait where all 5 of our species in our group of interest have the shared derived condition. Character number 2 now, it looks like only P and R have it. And so it can be sort of nestled right under the common ancestor of P and R. We can assume that the common ancestor had that condition, but nobody else on this cladogram have that. And same with trait number 3. But looking at trait 4 now, L, N, P, and R all have that. Well, conveniently, L, N, P, and R form a monophyletic group, and so we can subtend that node of the common ancestor of L, N, P, and R with trait number 4. Trait number 5 has a similar situation, but it's just shared with that small monophyletic group N, P, and R. But trait number 6 is a different one. This one we cannot form a monophyletic group with. And the only other thing that could have possibly happened if both N and R have this derived condition is it must have evolved twice. And that is how it's represented on the cladogram now. Changes? 7. One's older. Plain old N? Or the whole group that is P plus R? I'm not asking if P is older than N or if N is older than P. I'm wondering if the whole group that is PR, if we kind of put them into a single unit, P plus R. Well, the common ancestor between these two taxa is right there. And so actually they emerge at the same time, these two lineages. The lineage that led to N and the lineage that led to PR are the same age. So neither is older. That would sort of be the same as asking the question, which is older, P or R? Well, they're the same age. They have the same common ancestor. Moving on. Next, I'm asking for a synapomorphy for that small group that is P plus R. Trait number 2 and trait number 3. So the leaf shape and the plant habit are both synapomorphies for the group P and R. Is N plus R monophyletic? No. But what type of trait is trait 6? Even though it's found in N and R, it's not a synapomorphy. It's a homoplasy. All right, well, what's a sympplesiomorphy for the group NPR? Well, looks like we've got trait number 4 or trait number 1 would be a sympplesiomorphy for group NPR. And NPR is indeed a clade because it's all of the descendants of a single common ancestor. Now this last question, how many monophyletic groups do we have? There's one. Here's another one. There's a third. 4, and including the outgroup, we have 5 total monophyletic groups. In other words, we have 5 clades shown on this cladogram. Good work. All right, you ready for more? We're going to be building. We've been scaffolding this, and now we're ready to take it to the next level, I think. We've been using morphological characteristics for this whole virtual lab, and now we're going to take it to the next level. We're going to use DNA evidence. And here's what that's going to look like. Whoa, we've got a lot of information here. It looks kind of arbitrary. We've just got letters, and we don't know if they represent like red or 4 or 5. We don't know what they represent. All they are letters. And so let's talk about what these letters mean really quickly and kind of the basis of what DNA sort of is in a 10-second nutshell. This is how all DNA is set up. This is the same for all organisms that have DNA, which is all organisms, pretty much. Nucleotides are represented by these letters A, T, C, and G, and that's because there are just 4 nucleotides for the most part. And these stand for A is adenine, T is thymine, G is guanine, and C is cytosine. And 3 nucleotides together make up a single amino acid. All right, and so each genome and every single cell of all organisms, they have millions or billions of nucleotides, and they have tons of amino acids that are produced from these nucleotides. Well, when you have a string of amino acids, that's what produces a protein. And proteins are what make everything go round, what make everything alive. Proteins are sometimes enzymes, and they are what build a body and a cell. Proteins are very important. And one example would be like a pigment. You know, maybe there's a protein that codes for red pigment in a petal, and it's coded for by amino acids, which are coded for by different nucleotides. And it's a beautiful thing. Well, that's all you really need to know for this cladogram building exercise, really. But there's much more to know about DNA. But notice here we've got those same species, A, L, N, P, and R. Across the top are the nucleotide positions. And so looking at nucleotide position 1, notice they all have A. Out of all 4 letter options, there's no variation here. And so this is actually not a useful trait when we're trying to build a cladogram. It's not showing any variation, and so it wouldn't make any groups. And that is as opposed to this other type of trait, namely trait 19. There is at least one variation. There's one difference between the taxa here. And so that potentially could be useful for building a cladogram. And so really we only care about the informative traits, and that's the one that I've circled here in green with the check mark. These are informative because there's at least one or more taxa that have a different nucleotide than some of the others. Now, ideally, they have multiple differences. Just a single change is actually less useful, but still, it works. And so let's just extract those nucleotide positions that are informative to us. Let's get rid of all of the uninformative ones. And now we're left just with those 6 informative nucleotide positions. These are our characters. Okay, the characters are the nucleotide positions. The character states are the nucleotides themselves that are present at each nucleotide position. And since this is just a matrix of characters and character states, we have done this before. We can turn the outgroup, the character states of the outgroup into zeros. And we can turn the character states that are different from the outgroup into ones. And so that's exactly what I've done here. Like, for example, let's look at nucleotide position 20. The outgroup had a T, and so we turned that into a 0. And so all of the other taxa, A, P, and R that had nucleotide T in nucleotide position 20, they get a 0. While the 2 that had anything else, they had some other different nucleotide, then they'll have the derived condition of 1. And so that's what L and N have for nucleotide position 20. Okay, so now you can see how the 0s and 1s are converted from the A's, T's, C's, and G's. And so let's use this matrix of the characters and character states to build a cladogram. The outgroup is going to be the lineage of the most ancestral common ancestor on this cladogram. And so now we need to find the taxon that is the most similar, that has the fewest derived conditions from that outgroup, and we can assume that it is the one that branched off first. And it appears as though species A is the one that branched off first here because it had 5 ancestral conditions, while the others had fewer than 5 ancestral conditions. Let's see if we can figure out the rest of these. It looks like L has 3 features in common with A. N has 2 features in common with A. But P and R actually both have 4 features in common with A. And so since they're sort of tied, well, maybe there's something to this. Maybe they are closely related to each other. Notice that those features that they have, they're very, very similar to one another. In fact, they don't have any differences. And so the 2 of them are probably going to be in a small monophyletic group together, but it also appears as though they might be from a lineage that's not too far off from the common ancestor of A and the rest of these taxa. So it might look something like this, where we've got both P and R together on this one lineage that's close to A. And we had evidence for that because P and R were similar to A, but also were identical to each other. But P and R, it doesn't matter which order we place them in up here because, remember, we can pivot at any node. The left and the right doesn't make a difference here. And now that means we're only left with 2 others. Since we've done A and P and R, we just have L and N left. Compare L and N, we notice that they have almost all the same exact features. And so, again, that's evidence that they're probably going to be in a monophyletic group. It's going to be more likely than them being more closely related to any of the other taxa on this matrix, at least. So we've got L and N sort of finishing up the cladogram here, closely related to one another. But we do sort of have a little bit of an issue with one trait because it looks like trait number 30 is found in 2 taxa that are really not that closely related. So now let's get back to that in a second, though. We're going to trace all of these traits onto our cladogram so that we can count the total number of changes as is needed in a parsimony analysis. And so notice that trait number 19, it's shared amongst the large monophyletic group that includes all of our species in the group of interest. And so it will be placed right here. Now, We've been calling it trait number 19. It is nucleotide position number 19, technically. And what happened, if we look at the original matrix, is that nucleotide C changed to A. And so if we wanted to be more specific with that, we could, just to get more details onto this one cladogram. There are actually no traits that are derived for R, P, L, and N besides A. And so we actually might not have a synapomorphy for that group. So instead, let's look at these other traits. It looks like trait number 27 is definitely shared between R and P and nobody else on this cladogram. So that's evidence that R and P are a monophyletic group. They're the only two that have that derived condition. But N also shares the derived condition for character number 30. And so we can't put it as a synapomorphy under R and P since N has it as well, unless that exact trait evolved twice. Trait 30, after we get through the rest of these that are a little more straightforward. Nucleotide positions 20, 25, and 26 are derived for L and N and for no other taxa, and so those are going to be straightforward, best placed right there under the common ancestor that is shared between L and N. Now back to trait 30. We're pretty confident with this cladogram, and if it is true, then there's really only one way that character 30 could have evolved twice here, and that would be on the way up to RP and again on the way to N. So actually, even though it's DNA, we had an instance of convergent evolution here. One other thing that we could have done with trait number 30, and that would have to be represented on essentially a different tree topology. And that would be if trait 30 was actually a synapomorphy for the group that is RPN. That would mean that we could place it like right here on a cladogram. As long as R, P, and N were those final three lineages off of that smallest clade on this cladogram. And if this was a synapomorphy, if trait number 30 was actually a synapomorphy, well, then in order to trace all of these other traits out onto our cladogram, it's going to look something like this. Those three synapomorphies from our original cladogram that were subtending the group LN, those would now have to be homoplasies on this other cladogram where trait 30 was our synapomorphy. Because L and N don't form a monophyletic group in this other cladogram, the one that's represented on the left here. And so they would have had, those traits would have had to evolve independently twice. And so now the ultimate test here for a parsimony analysis is to compare the total number of changes on different possible cladograms here. And it looks like this one on the right that we built together, that one had 7 total changes. While the one on the left, when we include nucleotide position 19, this one has 9 total changes. And so it's less simple. There's more homoplasies, so it's harder to believe. The big one on the right with 7 total changes, that is the winner here. That is more parsimonious. That is the most parsimonious out of all possible options of tree topologies for these 5 species. All right, friends, that's all I've got for you for right now. Thank you so much for following along. I hope you've learned a lot about cladograms and parsimony analyses. I'll see you at the next video.